Hey there, ladies and gentlemen, and welcome to the second video for Chapter 13, How Populations Evolve. Let's look a little more closely at that actual mechanism of evolution and how it works. The major mechanism of evolution is called natural selection, but it can only function if there's a little bit of difference amongst the population to begin with. Remember, natural selection is a mechanism whereby whatever pressure is being applied to a population of a species that makes it harder to survive helps push change in that population in a certain direction. Natural selection and evolution are not a conscious process, but neither are they random, because it is those that survive the challenge and can breed, reproduce, that are able to pass down their traits. Not random. However, in order for there to be enough genetic variation in a population so that some will hopefully survive, you have to have some variation in the first place. We have two major ways of getting genetic variation in a population. This is either by random mutation. It happens sometimes. It can happen a little more commonly in DNA than you might think, just in the course of fertilization and genetic recombination, which also leads us to the other way that we get genetic variation, through sexual reproduction. Every single time new individuals are produced through sexual reproduction, we have an individual with a brand new combination of DNA. That's a brand new combination of all genes that's never been seen before. And this gives rise to new change, a new variety, so that hopefully someone in the population might survive. When we look at evolution functioning, speaking of which, we're not looking, at evolution occurring in terms of one individual's life. Evolution takes far, far too much time. Also, the variation survival of one individual is not enough to make change permanent in a species. Now, we're looking at change over the course of a population's existence, and not just one lifetime, but many, many generations. A population is a large enough group that some of those changes might actually become permanent. And we're specifically looking at which genes survive in the gene pool of the population. The gene pool is the variety of all the possible genes that individuals in that population might have. When we see changes in the gene pool over the course of many generations, this is the first sign of evolution. It's the earliest sign. Maybe those changes will be permanent, maybe they won't, and because it's so early in the evolutionary process, we call this a microevolution. Little guy. When we study evolution, part of what we're looking at is the frequency with which we see specific genes, particular alleles, particularly and specifically dominant, recessive, and heterozygous individuals. We have a specific system that we use to look at this, at these frequencies. This is called the Hardy-Weinberg principle, or Hardy-Weinberg equilibrium. The Hardy-Weinberg equilibrium, pardon me, maintains that if you see no selection in a population, meaning everything is perfect, everything is fine and dandy, there is no reason to have to change, survival is easy, no disease, plenty of food, no predators, good to go, and you see no mutation, and there is no migration, so someone who's born in one population stays in that population. They never leave. They never move. And you get large populations and random mating, and everybody can mate. All of the frequencies between homozygous dominant, heterozygous, and homozygous recessive alleles should stay fairly even. There's no reason for one of them to survive more successfully than any of the others. Of course, this is never what happens in reality, because these 5 conditions are never actually met in reality. But it is a mathematical principle that we can build from. That's our control group. See if everything was perfect. The equation for calculating the Hardy-Weinberg equilibrium is as follows. 1, aka the entire population, equals p squared, which is the frequency of homozygous dominance, plus 2p, which is the frequency of heterozygotes, plus q squared, which is the frequency of homozygous recessives. By knowing the frequency of even just one of these types of individuals, you can figure out the others. And this gives us an idea of what direction the population is being pushed in, because we can calculate which type of individual becomes most common in a population. This becomes very useful in health science when we look at the frequency of certain genetic diseases, particularly things like phenylketonuria, which is the ability to break down a particular acid. But also any other genetic inherited disease. We can watch and track its progress through a population and find out how dangerous or how severe it may be by how widespread it is. And also how likely we are to have new patients, which tells us how carefully we should be on the lookout for someone with this disease by seeing how widespread it becomes in a population. Alright folks, this is going to wrap it up for the second video of Chapter 13, and I will see you next time. Bye folks.